Welcome back to the deep dive. Today, we are taking a full, comprehensive journey into clinical immunology.
Mm,
And I don't just mean the textbook definition, but the actual messy, real-world function of your body's, well, its most complex defense system.
it really is. We're talking about this multi-layered network of cells, organs, and these specialized soluble proteins that are constantly running surveillance.
And defense operations, all happening inside you right now.
It's a crucial topic because at the end of the day, the immune system touches absolutely everything. Our mission today is to give you a genuine shortcut to being well-informed about your own defenses.
Okay, so where do we start?
We'll decode that essential distinction between the fast, sort of generalized innate immune system and the slower, much more sophisticated adaptive system.
Right.
But more importantly, we'll explore the clinical consequences of when these systems fail.
Which is where it gets really interesting.
Exactly.
Yeah.
Everything from allergy and devastating primary deficiencies to, the revolutionary new methods used to harness immune power in cancer therapy and transplantation.
And when you look at the history of this field, it's just astonishing how recently we began to grasp its complexity.
Oh, it is.
For centuries, people just observed that if you survived the plague, well, you were immune to the plague. That concept of immunity was there, but the idea of an actual system.
A functional, defined network.
Yeah, that wasn't formally established until, the late 19th century?
That's right. It moves from just a simple observation of protection to understanding it as this integrated functional anatomy.
And now we know it governs protection, yes, but it's also fundamental to tissue repair.
Wound healing, and when it's dysregulated, it is the root cause of an immense variety of diseases that potentially involve every single organ system in the body.
It truly is the ultimate functional anatomy.
It is.
Okay, let's unpack this system. I think the best way is to view it through the lens of its two main functional components.
Innate and adaptive.
Right. And these components don't operate independently. They're constantly communicating, largely using a shared vocabulary of signaling proteins we call cytokines. Let's start with the immediate, generalized defense, innate immunity.
Think of innate immunity as the rapid response team.
The first responders.
Exactly. Or, the biological alarm system. It is designed for speed, providing protection, often within minutes of encountering a threat. Its massive advantage is its pre-preparedness.
But it must have a downside.
It does.
Yeah.
Its massive limitation is its lack of specificity.
So it doesn't care what the threat is, just that it's a threat.
Precisely. It recognizes these broad generic molecular structures, we call them pathogen-associated molecular patterns, that are typically conserved across classes of microbes, but are absolutely not found in your own mammalian tissue.
So it's a simple us versus them recognition.
A very effective one. And because the recognition is generic, any specific stimulus, say a certain bacterial cell wall component, will elicit essentially the identical response across different individuals. It's hardwired.
Now contrast that immediacy with the adaptive or acquired immune system.
The adaptive system is the specialized forces. It takes time, often days or even weeks, to mobilize.
But the payoff is worth it.
The payoff is immense. It offers exquisite specificity. It can discriminate between molecular structures that differ by only a few atoms. It's also incredibly flexible, capable of mounting a response to a virtually unlimited number of different molecules.
And the adaptive system gives us the ultimate defense feature, memory.
That's the key. Immunological memory is what confers long-lasting protection. Once the adaptive system has been trained against a pathogen, the second encounter results in a response that is much faster, much stronger, and vastly more effective.
Which is the entire biological premise behind vaccination.
It's exactly that.
Before either of these forces are truly engaged, though, we have the most foundational defense line of all. Our physical and commensal barriers.
The walls of the castle.
Yes, the defenses that are always on, providing passive structural protection.
They are the first line, and honestly, often the most successful. Take your skin, for instance. It's more than just a physical wall of keratinized cells.
It's an active barrier.
It's a hostile environment for microbes. It actively limits colonization through its low pH, its hydrophobic sebaceous oils, and it even secretes enzymes like lysozyme and these crucial antimicrobial peptides called defensins.
And the vast, delicate surfaces of the mucosal membranes, which are constantly exposed, they must have an even more sophisticated layered defense.
They absolutely do, particularly in the respiratory, GI, and genitourinary tracts. They secrete this thick mucus to physically trap invaders.
So it's like flypaper?
Kind of, yeah. And they use enzymes like lactoferrin, which actively binds and starves invading bacteria of iron,
Yeah.
and lysozyme. They also leverage a secreted form of the adaptive component, immunoglobulin A, or IgA.
IgA.
Its primary function is to prevent pathogens from ever attaching to the epithelial cells in the first place.
And let's not forget the crude but highly effective mechanical maneuvers we often take for granted.
Right. I mean, think of the continual beating of the cilia in the respiratory tract, pushing mucus and trapped particles upward.
Combined with coughing and sneezing.
Exactly. Expulsion mechanisms. In the stomach, you have hydrochloric acid, and throughout the intestine, the constant muscular action of peristalsis and, of course, the ability to vomit are essential clearance methods.
Here's where the system truly reflects a deep biological partnership. The crucial role of the, 10 to the 14th power commensal bacteria residing at those epithelial surfaces, our microbiome.
This relationship is absolutely fundamental to our defense. The commensal flora provide a constitutive defense simply by occupying real estate.
They're taking up space.
They're taking up space, and they compete aggressively with pathogenic microorganisms for scarce nutrients. They also produce useful metabolites like fatty acids and bactericidins that actively inhibit the growth of many pathogens.
But the source material also highlights a vital regulatory role for these microbial communities.
Yes, they aren't just passive competitors. They actively help shape the immune response itself, especially in the gut. By signaling with our immune cells, they induce specific populations of regulatory T cells, which are critical for preventing inappropriate inflammation.
So they're keeping the peace.
They are, keeping the system in balance in a region that's constantly exposed to foreign material.
Which leads directly to that moment that illuminates their importance. The risk of opportunistic infection when that normal flora is decimated.
That's the aha moment.
Yeah.
It shows you how vital this symbiosis is. When you strip away the normal competitive flora, say, to the use of broad-spectrum antibiotics, you immediately create an undefended ecological niche.
And something will fill that vacuum.
Something will. And the textbook example, a major clinical concern, is the rapid colonization and opportunistic infection by organisms like Clostridioides difficile,
C. diff.
which can cause life-threatening diarrhea. It proves that the microbiome is indeed a crucial pillar of immune defense.
Okay, so if those barriers fail, the innate system mobilizes its cellular heavy hitters, starting with the group we call phagocytes.
Literally meaning eating cells.
Right. What are their central roles in the immediate response?
Their roles are twofold. They're the cleanup crew and the signaling hub. Phagocytes are specialized cells that ingest and kill microorganisms, but they also scavenge cellular debris following injury.
Okay.
And importantly, they are crucial for producing the early inflammatory molecules that regulate the mobilization of the rest of the immune system.
And this includes the three main types.
Yes, you have the neutrophils and monocytes circulating in the blood, and then the tissue-resident macrophages.
And macrophages are adapted to their local environments, right?
Exactly. We call them Kupffer cells in the liver, alveolar macrophages in the lung, and microglia in the central nervous system. They are the fixed guard posts of the tissues.
And their ability to recognize an invader without ever having seen it before, that hinges entirely on pattern recognition. How does that system work?
It's elegant in its simplicity. Phagocytes express what are called pattern recognition receptors, or PRRs, which are designed to scan for pathogen-associated molecular patterns, or PAMPs.
PAMPs.
PAMPs are those signature molecular motifs that are not found on mammalian cells. Things like lipopolysaccharide or LPS from gram-negative bacteria, peptidoglycans from cell walls, or unique structures in bacterial DNA or viral double-stranded RNA.
So once a PRR recognizes a PAMP, what is the immediate non-negotiable activation pathway? This seems to be the trigger for inflammation itself.
It is the point of no return for acute inflammation. The interaction triggers two critical parallel responses inside the cell. The first is the activation of NFE.
Nuclear factor kappa B?
Right, a master regulator that translocates to the nucleus and just flips the switch on hundreds of pro-inflammatory genes.
And the second pathway involves structures with an incredible name, the inflammasome.
The name fits its function perfectly. This involves another class of receptors, the NLRPs. When these are triggered, and not just by microbial components, sometimes by internal danger signals like the uric acid crystals that cause gout, they complex with other proteins to form the inflammasome.
Why is the inflammasome activation so clinically relevant?
Because it acts like a molecular executioner. It leads directly to the maturation and rapid excretion of interleukin-1 beta, IL-1, which is one of the most powerful pro-inflammatory cytokines known.
So it's a huge alarm bell.
A massive one. And this direct link explains the pathogenesis of several intense inflammatory disorders, including acute gout attacks and some rare severe familial fever syndromes, where excessive IL-1 production is the defining feature.
So understanding this activation point gives us a direct drug target.
Exactly.
Moving specifically to the most prolific phagocyte, the neutrophil. These are the shock troops you mentioned. Just how massive is their production rate?
It's astonishing. They're produced at a rate of about 10 to the 11th power cells daily,
Every day.
but they are highly sacrificial. They have a short half-life of only 6 hours. They get recruited rapidly to the site of infection by chemical signals like IL-8, and once they're in the tissue, they're just dedicated to ingestion and killing.
And they employ two distinct mechanisms to kill the microorganisms they ingest.
Correct. The first is oxidative killing, often called the respiratory burst. This is catalyzed by the NADPH oxidase enzyme complex.
Okay.
It rapidly consumes oxygen to produce highly reactive oxygen species like hydrogen peroxide and superoxide. These radicals are lethal to microorganisms inside the phagolysosome.
And the second mechanism is non-oxidative killing.
This relies on the massive arsenal of preformed enzymes stored in the neutrophil's granules.
So it's got its weapons ready to go.
Absolutely. Lysozyme, collagenase, and crucially, myeloperoxidase, which uses hydrogen peroxide to produce hypochlorous acid, essentially bleach, giving them broad spectrum coverage against bacteria and fungi.
Beyond ingestion, there is that remarkable recent discovery about how they deal with larger threats, which you alluded to earlier, notosis.
Neutrophil extracellular trap formation, or NETs. It's truly one of the most fascinating aspects of innate defense. The activated neutrophil sacrifices itself, releasing its entire nuclear contents, its chromatin, into the extracellular space.
So it just explodes.
It decondenses and shoots out its DNA, which is loaded with those granule proteins like elastase, forming this sticky, net-like mesh.
Wow.
And this net physically immobilizes or kills microbes outside the cell, trapping them and preventing their spread.
And what we commonly see as a sign of infection is directly related to the death of these cells.
Absolutely. This is not just a sign of infection. It is literally the battlefield debris.
So it's dead neutrophils.
It's the accumulation of dead and dying neutrophils and the digestive enzymes they release, which degrade the surrounding tissue. If it's extensive, this forms an abscess.
So we've covered how the phagocytes fight, but their ability to target those invaders relies heavily on a system of flagging. Can you explain how opsonization essentially paints a target for the phagocytes?
Yeah, that's a great way to put it. Phagocytes can sometimes engulf pathogens on their own, but engulfment is drastically enhanced by opsonins, which literally means to prepare for eating.
Okay.
These soluble molecules, like C-reactive protein from the liver, the complement fragment C3b, and antibodies, they act as a molecular bridge. They bind to the pathogen on one end and to specific receptors on the phagocyte on the other, facilitating rapid destruction.
Opsonins are a form of communication, but the entire immune response is regulated by this massive network of signaling proteins, the cytokines. Given that more than 100 have been identified, we have to focus on the two crucial intracellular signaling pathways that govern cellular behavior.
We can definitely simplify it to two central pathways. We already mentioned MC, which is activated by TNF and PRRs.
Right.
When a receptor is bound, it initiates the IKK complex. This complex destroys the inhibitor of kappa B, freeing enough food to enter the nucleus and activate gene transcription.
Turning on the inflammatory cascade.
Precisely. And the second major pathway is JAK-STAT.
The Janus kinase signal transducers and activators of transcription pathway.
A mouthful, I know. It's used by critical cytokines like IL-2, IL-6, and IFN-gamma. When the cytokine binds its receptor, it recruits and activates these JK proteins.
And the JAKs activate the STATs.
The JAKs then phosphorylate STAT proteins, which dimerize, enter the nucleus, and activate transcription.
This sounds incredibly abstract, but here is the perfect example of molecular biology translating directly into clinical medicine.
It's the foundation of modern immunomodulation. By understanding these specific intracellular steps, pharmaceutical science has developed small molecule inhibitors. The Janus kinase inhibitors like tofacitinib, baricitinib, and upadacitinib.
The JAK inhibitors.
Yes, these oral drugs block the signaling within the cell, effectively muting the inflammatory response driven by IL-6 and IFN-gamma. And they are now standard treatments for severe rheumatoid arthritis and inflammatory bowel diseases.
Let's discuss the final major pillar of the innate system, the complement cascade. This is described as a tightly regulated but rapidly amplifying biological defense mechanism.
It is indeed an explosive cascade. It involves over 20 proteins, mostly produced in the liver. They circulate in an inactive form, and when triggered, they rapidly activate each other in a sequential, amplified manner.
Like a fire alarm system.
That's a great analogy.
When we learn that this cascade can be triggered in 3 distinct ways.
Exactly. The alternate pathway is the most ancient and direct, activated by C3 binding directly to microbial cell wall components like LPS.
Okay.
The lectin pathway uses mannose binding lectin to recognize microbial carbohydrates, bypassing the need for antibodies. And finally, the classical pathway is initiated by the adaptive system, when 2 or more IgM or IgG antibodies bind to an antigen.
And that classical pathway is naturally controlled by the C1 inhibitor.
C1, yes, that's crucial to remember later.
But regardless of the trigger, they all funnel into a single, highly effective outcome.
They converge on C3 activation, which leads to the assembly of the membrane attack complex, the MAC.
The MAC, C5 through C9.
Right. And this complex inserts itself directly into the pathogen's cell wall, creating pores that cause osmotic lysis. It literally punches holes in them.
And this step is particularly critical for defending against what kind of bacteria?
Encapsulated bacteria, like Neisseria species.
And beyond direct killing, the resulting fragments act as powerful regulatory molecules throughout the immune landscape.
They absolutely do. We mentioned C3b as an opsonin. Other fragments are powerful chemotactic agents, recruiting leukocytes to the site of inflammation. And others are anaphylotoxins, binding to mast cells and triggering the release of histamine.
Which increases vascular permeability and aids the infiltration of more immune cells.
You got it. It's all connected.
Now we shift to the adaptive system, which provides that specificity and memory. To understand how it works, we have to look at the structure of the lymphoid organs, the places where these cells are trained and where they meet their enemy.
Right, and lymphoid organs are defined by their function. You have the primary organs, the bone marrow and the thymus, which are the development and training centers.
The boot camps.
Exactly. B cells mature in the marrow, while the thymus is the site of stringent T cell selection, where any T cell that recognizes self too strongly is quickly eliminated. That process is called central tolerance.
And the secondary organs are the staging grounds where the action happens.
These are the lymph nodes, the spleen, and the MALT, or mucosa-associated lymphoid tissue. These sites are specifically engineered to trap and concentrate foreign substances, and are the major locations where naive lymphocytes first encounter their specific antigens.
And the organization is very deliberate, right?
Especially in a lymph node. B cells cluster in the outer cortex, T cells and the dendritic cells, the master presenters, reside in the inner paracortex, and the medulla is where the antibody-secreting plasma cells take up residence.
Let's start with humoral immunity, centered on B cells and the production of antibodies. What has to happen for a B cell to go from quiet patrol to full-scale antibody factory?
Okay, so each B cell expresses a unique B cell receptor on its surface. When it encounters its matching antigen and critically receives the necessary co-stimulatory signals from helper T cells.
It's not a solo act.
Not at all. It needs that T cell help. Once it gets it, it undergoes 2 dramatic steps. First, rapid clonal expansion, creating an army of identical cells. Second, somatic hypermutation.
Which sounds intense.
It is. It fine-tunes the B cell receptor genes to generate populations with even greater affinity for the antigen. It's like a rapid evolution in real time.
The result of this process is the engine of long-lasting immunity.
The B cells differentiate into either antibody-producing plasma cells, the short-term factories, or into long-lived memory B cells. These memory cells can patrol the body for decades, ready to be reactivated instantly upon re-exposure.
Which is the physical manifestation of immunological memory.
Exactly that.
Antibodies, or immunoglobulins, are the product. We hear about the 5 main classes: IgG, IgA, IgM, IgE, IgD. How does the body use these different classes?
The class, or isotype, is determined by the heavy chain. IgM is typically produced first in a primary response. IgA, as we mentioned, is specialized for mucosal surfaces. And IgG is the dominant high-affinity antibody of the secondary memory response. IgG is particularly versatile. It facilitates opsonization, it helps NK cells kill, a process called ADCC.
Antibody-dependent cellular cytotoxicity.
Yes, and it activates the classical complement pathway.
Speaking of memory, that distinction between the primary and secondary response is the foundation of vaccine efficacy.
It is the defining qualitative difference. The primary response is slow. It takes 5 to 10 days to generate protective levels, and it's dominated by low-affinity IgM.
But the secondary response
Is a biological rush. The lag time shrinks dramatically, the total quantity of antibody is much higher, and the response is dominated by high-affinity IgG. The memory cells are so primed that the response often doesn't even require the full T-cell help that the primary response demanded.
Now to cellular immunity, mediated by T lymphocytes. The fundamental difference here is that T cells cannot recognize antigens floating freely in the body like B cells can. What is the essential requirement for T cell activation?
T cells are highly regulatory and suspicious. They cannot recognize an intact native protein.
So it has to be processed first.
It must be. It has to be broken down into component peptides and presented on the surface of an antigen-presenting cell, or APC, in association with highly specific HLA molecules.
And the T cell receptor only recognizes the complex of peptide plus HLA.
That's the key. No HLA, no recognition.
T cells are divided into two main subgroups, based on the HLA molecules they recognize. Let's start with the cytotoxic killers, the CD8 plus T cells.
CD8 plus T cells recognize peptides presented on HLA class I molecules HLA-A, B, and C.
And class I is on almost all our cells.
On nearly all nucleated cells in the body, so their role is to survey for internal abnormalities, viruses, or cancer. When they find an infected cell, they release perforin and granzymes, which induce rapid apoptosis.
So they tell a cell to commit suicide.
Essentially, yes, killing the target cell without creating widespread inflammation.
And the regulatory orchestra conductors, the CD4 plus T cells, the helper cells.
They recognize peptides presented on HLA class II molecules DR, DP, DQ, which are predominantly expressed only on professional APCs.
Okay, so a more limited audience.
A very specific one.
Right.
Their role is immunoregulatory. They produce massive amounts of cytokines to support CD8 plus activation, drive B-cell differentiation, and activate phagocytes. The source material details critical subsets based on their cytokine output: Th1 for intracellular pathogens, Th2 for allergy, and Tregs for suppression.
Given the power of these T cells to destroy, their activation must be highly regulated. This balance between activating and inhibitory signals is now the focus of modern medicine.
It is the delicate balance of life. T-cell activation requires two signals: the T-cell receptor binding the HLA complex, plus a co-stimulatory signal like CD28 on the T-cell binding to CD80 or 86 on the APC.
But the body also uses inhibitory signals, brakes.
Brakes, exactly. A great example is the inhibitory molecule CTLA-4.
And CTLA-4 has been directly translated into targeted anti-inflammatory therapy.
Yes. CTLA-4 naturally competes with CD28 for binding to those co-stimulatory molecules. When CTLA-4 wins, the T-cell is inhibited.
So the drug abatacept
Is a fusion protein incorporating CTLA-4. By giving it to patients with rheumatoid arthritis, it effectively shuts down T-cell activation, mitigating the autoimmune destruction.
But the pathway that has dominated the headlines in the last decade is the PD-1 pathway, especially in oncology.
The programmed cell death 1, or PD-1 receptor, is another critical inhibitory molecule on lymphocytes. When it binds its ligand, PD-L1, which is expressed on APCs, endothelial cells, and critically, many cancer cells.
It sends a turn-off signal.
The very strong one.
Yeah.
And cancer cells co-opt this pathway to evade the immune system.
This sounds like the perfect target for therapy.
It is. Monoclonal antibodies that block this interaction, the immune checkpoint inhibitors like nivolumab, effectively take the brakes off the T cells, allowing them to attack the tumor aggressively.
And this is now a cornerstone treatment for cancers like melanoma and lung cancer.
It is, demonstrating the profound systemic effect of manipulating these molecular regulators.
When this highly regulated system is activated, correctly or incorrectly, the first clinical result we see is inflammation. We all recognize the classical external signs: heat, redness, pain, and swelling.
And those signs are the external manifestation of immediate vascular changes driven by inflammatory mediators.
Like what?
Leukotrienes, prostaglandins, histamine, kinins, and those complement anaphylotoxins we mentioned. They act rapidly to cause massive vasodilatation and increase vascular permeability,
Which allows fluid and immune cells to flood the affected tissue.
causing the heat, swelling, and redness we observe.
But the response is not just local. There's a massive systemic change known as the acute phase response.
This is driven by the systemic release of key pro-inflammatory cytokines, specifically IL-1, TNF-alpha, and IL-6, which are produced at the local injury site.
And they act on the brain and the liver.
Right. On the hypothalamus to cause fever, and critically, on the liver to stimulate the production of acute phase proteins.
How significant is the scale of this liver response?
It can be huge. Circulating levels of C-reactive protein, or CRP, can increase 100 to 1,000-fold in a matter of hours.
Wow.
And these proteins are defense mechanisms. CRP is an opsonin, for example. But the liver also increases production of fibrinogen for wound healing and iron-binding proteins to try and starve invading bacteria.
If that inflammatory response spirals out of control, we enter one of the most dangerous clinical scenarios: septic shock. This is overwhelming, uncontrolled systemic inflammation.
Septic shock is the ultimate failure of regulation. It's characterized by massive runaway cytokine production leading to widespread vascular damage, severe hypotension, and hypovolemia.
And the clinical urgency here is driven by the vascular damage itself.
It is. Uncontrolled neutrophil activation within the blood vessels releases proteases and oxygen-free radicals that just shred the vascular endothelium.
And this damage triggers a dangerous clotting cascade as well, doesn't it?
It does. The endothelial damage directly activates the coagulation pathway, leading to clotting within damaged vessels throughout the body, a condition called disseminated intravascular coagulation, or DIC.
And septic shock is frequently caused by gram-negative bacteria.
Yes, because their lipopolysaccharide, their LPS, is a potent PMP that triggers a particularly aggressive inflammatory response through those PRRs we discussed.
Let's shift to diagnosis. To monitor active inflammation, clinicians rely heavily on two key markers, CRP and ESR. It's important to understand why both are used and the nuance between them.
They both measure inflammation, but they do it very differently.
Yeah.
C-reactive protein, CRP, is the direct biomarker of acute inflammation.
It's made by the liver.
Synthesized by a liver, rises rapidly within just 6 hours of the inflammatory stimulus, and it has a very short half-life, only 18 hours.
It's a real-time indicator.
Exactly. That short half-life is what makes sequential CRP measurements so useful for monitoring therapy. If treatment is working, the CRP level drops promptly.
And the erythrocyte sedimentation rate, or ESR, is a far more indirect measure.
Correct. ESR measures the rate at which red blood cells fall through plasma, and that rate is determined by the plasma protein composition.
How does that work?
Red blood cells normally repel each other because they are negatively charged. However, positively charged plasma proteins, primarily fibrinogen and immunoglobulins, neutralize this charge.
And that causes them to clump.
Right, they form stacks called rouleaux, which sediment much faster. So a high ESR means high levels of these proteins.
So here is the crucial clinical distinction. While CRP is generally preferred for acute inflammation, there are conditions where the results diverge, and this divergence offers critical diagnostic clues.
That divergence is key. You might see a high ESR with a normal CRP in conditions that cause massive immunoglobulin production, like multiple myeloma.
Why is that?
Because immunoglobulins are not acute phase reactants, so they raise the ESR, but they don't trigger the acute CRP spike.
I see.
Conversely, some systemic autoimmune diseases, like systemic lupus erythematosus, or SLE, can be actively inflamed with a high ESR, but their CRP may remain only minimally elevated. So in those specific cases, ESR is actually the better marker.
Moving on to the failure of the system itself, primary immune deficiencies, or PIDs. These are usually genetically determined defects, typically presenting in childhood or adolescence.
And the consequences are threefold: recurrent infections, paradoxically common autoimmunity, and an increased susceptibility to viral-driven malignancy, like EBV-associated lymphoma.
And the clinical pattern of infection almost always tells you which specific component of the immune system has failed.
Exactly. Each component fights specific pathogen classes, so the type of infection is a huge clue.
Let's start with defects in the rapid response T phagocyte deficiencies, which typically manifest as severe bacterial and fungal infections.
The most dramatic example is chronic granulomatous disease, or CGD. This is a genetic failure in the NADPH oxidase enzyme complex.
The one that causes the respiratory burst.
Right. It results in the complete failure of oxidative killing. These patients are uniquely susceptible to organisms that produce the catalase enzyme, which breaks down hydrogen peroxide. We are talking about organisms like Staphylococcus aureus and Aspergillus mold.
And clinically, they form granulomas.
Yes, because the immune system tries desperately to wall off the infection it can't kill.
And the other severe phagocyte defect, leukocyte adhesion deficiencies. This has a very striking and specific clinical presentation.
It does. These are defects in adhesion molecules, most famously involving the integrin LFA1. If this molecule is defective, the neutrophils in the blood cannot bind to the endothelial wall and cannot exit the bloodstream to reach the site of infection.
So the core clinical clue is what isn't there at the site of infection.
Exactly. The patient has recurrent, life-threatening bacterial infections, but the site of infection will classically lack pus formation.
Because the neutrophils can't get there.
They physically cannot infiltrate the tissue. And ironically, if you take a blood sample, the peripheral neutrophil counts may be extremely high because they are trapped in the circulation. That lack of pus is a critical differentiator.
Now, for the most common PIDs, antibody deficiencies, characterized by recurrent infections of mucosal surfaces, typically with encapsulated bacteria.
The most common PID of all is selective IgA deficiency. It affects about 1 in 600 Northern Europeans.
And since IgA is for mucosal defense,
People with this deficiency may have recurrent, mild respiratory or GI infections, but, many are completely asymptomatic and we rarely need to treat it specifically.
but then you move to the more severe and often adult onset form, common variable immune deficiency, or CVI.
CVI is a highly variable deficiency defined by low serum IgG and the inability to mount an adequate antibody response to vaccines or pathogens.
So these patients get chronic infections.
Chronic, debilitating infections that lead to progressive end organ damage, often bronchiectasis in the lungs. What makes CVI complex is the high prevalence of associated autoimmune diseases like ITP and an increased risk of specific lymphomas.
The management for these patients is standardized and lifelong.
Yes, the mainstay is immunoglobulin replacement therapy, either IVEG or subcutaneous. This is passive immunity.
You're just giving them the antibodies they can't make.
Exactly. Pooled human IgG. It must be administered lifelong, aiming to maintain adequate TRAF levels to prevent irreversible organ damage. And importantly, all patients with PIDs must avoid live vaccines.
Finally, the most devastating category: primary T lymphocyte deficiencies. Given the T cell's central regulatory role, these deficiencies compromise the entire immune system.
They compromise everything. The most severe is severe combined immune deficiency, or SCID, the bubble boy disease.
Right.
It involves a catastrophic failure of T cell maturation, often affecting B and NK cells too. Without a functional adaptive system, infants present soon after birth with recurrent, devastating infections from all classes of pathogens.
And without intervention.
It is rapidly fatal. It requires curative measures, either stem cell transplantation or, for specific genetic causes, gene therapy.
We should also mention DeGeorge syndrome.
Yes, which results from a genetic deletion on chromosome 22q11 that causes a failure of thymic development. This leads to a varying degree of T cell deficiency alongside other severe congenital problems like heart defects.
When the immune system turns against the host, we enter the realm of autoimmunity, the ultimate failure of immune tolerance. The immune system begins to recognize self-tissue as foreign.
And this happens when self-reactive lymphocytes manage to escape two critical control points. First, central tolerance, which is the elimination of self-recognizing cells during development.
And second, peripheral tolerance.
Which is the active suppression exerted by those T-regulatory cells, or Tregs, in the circulation.
We know that genetics, particularly specific HLA loci, play a huge role, but what environmental factors often pull the trigger?
Genetics sets the stage, but a non-self trigger is usually required. Infection is the most common external trigger, often via a mechanism called molecular mimicry.
What's that?
It involves cross-reactivity, where an antigen expressed by a pathogen happens to look structurally similar to a host protein. The immune response kills the microbe, but then accidentally targets the host tissue.
Can you give an example?
A clear one is Guillain-Barré syndrome, where T cells react to peripheral nerves following a Campylobacter infection.
To categorize the different ways the immune system causes tissue damage and autoimmunity, we rely on the Gell and Coombs hypersensitivity classification. Can you walk us through types 2, 3, and 4?
Sure. Type I is allergy, which we'll cover next. Type 2 is mediated by specific IgG or IgM antibodies binding directly to fixed antigens on the surface of host cells or tissues.
And that binding triggers destruction.
Exactly. It triggers complement or phagocytes, leading to cell destruction, as seen in autoimmune hemolytic anemia or Goodpasture's disease.
How does that compare to type 3?
Type 3 is the immune complex disease. Here, the antibodies bind to soluble antigens, forming circulating immune complexes. These complexes deposit in blood vessel walls or tissues, triggering the classical complement cascade and recruiting massive inflammatory infiltration.
And this causes systemic diseases.
Yes, like systemic lupus erythematosus, SLE, and cryoglobulinemia.
And there's a crucial diagnostic clue for type 3.
Yes. If the patient is suffering an active type 3 disease, the complement system is being heavily consumed by these deposited complexes, so circulating levels of C3 and C4 will typically be low.
Okay, finally, type 5e hypersensitivity, the delayed type response.
This is purely cellular mediated entirely by activated T cells and macrophages. It typically develops 24 to 72 hours after exposure.
So things like contact dermatitis.
Right, but also autoimmune diseases where T cells directly attack tissue, such as type 1 diabetes and Hashimoto's thyroiditis.
Shifting to allergy, which is type hypersensitivity, this is an inappropriate IgE-mediated and immediate response to an otherwise harmless substance.
The mechanism is a biological setup. Initial exposure causes B cells to produce high levels of specific IgE antibodies.
And this IgE doesn't just float around.
No, it binds with high affinity to specialized receptors on mast cells in the tissues, so the mast cells become armed. Re-exposure to the allergen then acts as a molecular bridge, cross-linking these surface-bound IgE molecules.
And that's the trigger.
It's an instant signal that triggers massive mast cell degranulation.
And that degranulation releases the mediators that cause immediate symptoms.
Exactly. Histamine, leukotrienes, and prostaglandins cause the immediate symptoms. Smooth muscle contraction, rapid onset of a runny nose or wheezing, increased vascular permeability, and, in the most severe cases, anaphylaxis.
The source material mentions the hygiene hypothesis to explain the rising global incidence of allergic disease.
This theory suggests that improved sanitation and reduced exposure to a wide variety of microbial antigens in early life, including dirt, bacteria, and infection biases, the developing immune system.
Oh, so?
Instead of robust Th1 responses, which are geared toward killing microbes, the immune system leans heavily toward Th2 responses, which are the main drivers of allergic disease and IgE production.
How are clinicians getting more accurate in diagnosing and risk stratifying allergies, especially with complicated foods like nuts?
Well, traditional diagnosis uses the skin prick test, which gives a rapid result but lacks precision, and you have to stop antihistamines. The modern advance is component-resolved diagnostics, or CRD.
And what does that do?
This uses purified recombinant allergenic molecules to test specific IgE against individual proteins within an allergen source.
What's the advantage of that increased detail?
CRD can reliably distinguish genuine primary allergy from mild, harmless cross-reactivity. For example, with a hazelnut allergy, if the test reveals IgE only against core A1, that suggests a mild oral allergy syndrome linked to birch pollen.
But if it's something else?
If the test reveals IgE against core A9 or core A14, these are heat-stable, severe reaction risk proteins. That confirms primary allergy and signals the urgent need for an epinephrine autoinjector prescription.
For investigating systemic reactions, what marker is critical in the acute setting?
Mast cell tryptase. When severe anaphylaxis occurs, the massive mast cell degranulation releases tryptase, which peaks in the blood 1 to 2 hours after the systemic reaction.
So it's a way to confirm anaphylaxis after the fact.
Precisely. It's key for confirming that an acute episode was, in fact, anaphylaxis.
Finally, we must make a crucial distinction concerning angioedema, the localized, non-pitting swelling that can be life-threatening if it affects the larynx. The treatment depends entirely on its mediator.
This is a classic differential diagnosis point. Angioedema can be histamine-mediated, meaning it's allergic, often associated with hives, and it responds well to standard antihistamines and steroids.
Where?
Or it can be bradykinin-mediated.
And the most common genetic cause of the latter is hereditary angioedema, or HAE.
HAE is an autosomal dominant disorder caused by deficiency in C1 inhibitor.
C1 again.
That's the one. Its deficiency leads to uncontrolled local production of bradykinin, causing severe pain and swelling. Crucially, HAE is never associated with urticaria or other allergy symptoms. The attacks often present as potentially fatal laryngeal obstruction or severe recurrent abdominal pain.
And because it's not histamine, standard allergy medication is useless.
Exactly. Management requires specific treatments. C1 inhibitor concentrate or bradykinin receptor antagonists like icatibant to halt the acute attack. The investigation clue here is that an acute HAE episode is accompanied by low C4 levels.
The ultimate stress test of the immune system's specificity is transplantation, where the goal is to introduce foreign tissue without triggering an aggressive immune response in the recipient. What is the central genetic challenge?
The central barrier is the human leukocyte antigen, or HLA, proteins. These genes are highly polymorphic, meaning they vary massively between individuals.
And because HLA molecules are the required antigen presenters for T cells,
any disparity between donor and recipient HLA antigens will be recognized as intensely foreign, triggering rejection.
the timing of rejection dictates the mechanism of damage. What is hyperacute rejection?
This is a catastrophic, rapid, irreversible destruction of the graft, occurring minutes to hours after transplant. It's a type 2 hypersensitivity mediated by preformed recipient antibodies against the donor's HLA.
But it's rare now.
Very rare, because we perform aggressive pre-transplant cross-matching to ensure the recipient doesn't already have antibodies against the donor.
The most common rejection seen today is acute cellular rejection.
This typically manifests between 5 and 30 days post-transplant and is mediated by activated T lymphocytes, a type IV hypersensitivity reaction.
And this is treatable.
It usually causes a drop in graft function, but importantly, it is often treatable with a temporary increase in standard immunosuppressive therapy.
But the source material notes that chronic allograft failure is the major cause of long-term graft loss.
Chronic failure is an insidious process, often associated with fibrosis and scarring in the graft's blood vessels. It results from a combination of ongoing low-grade immunological damage and non-immune factors like chronic drug toxicity or hypertension.
Speaking of drugs, long-term immunosuppression, while necessary for the graft's survival, comes with a major cost to the recipient.
The cost is increased vulnerability to infection and malignancy. By suppressing T-cell activity, we drastically increase the risk of opportunistic infections like CMV and pneumocystis.
And the malignancy risk.
The reduced immune surveillance leads to a significant increase in viral-associated tumors, particularly EBV-linked lymphoma, which we call PTLD, and HPV-linked skin tumors.
Let's transition to tumor immunology, the great paradox. Cancer cells survive by co-opting the very inhibitory pathways the immune system uses to maintain self-tolerance.
It's a remarkable feat of biological evasion. Cancer cells are highly mutated and should be targeted, but they learn to express molecules like PD-L1.
The ligand for PD-1.
Right, so when a T cell comes along, ready to attack, the cancer cell engages that PD-1-PD-L1 interaction, delivering that strong stand-down signal, effectively turning off the T cell response and evading destruction.
This realization led directly to the breakthrough of immune checkpoint blockade.
This therapy strategically blocks these downregulators. We use monoclonal antibodies that target CTLA-4 or more commonly PD-1 or PD-L1.
You're taking the brakes off the T cells.
Exactly. And by removing these molecular brakes, we unleash the T cell's inherent anti-tumor power. This has revolutionized the treatment of previously intractable cancers.
But as you pointed out with immunosuppression, there is always a trade-off when you dial up immunity so aggressively.
Absolutely. The price of enhanced anti-tumor immunity is the development of serious inflammatory side effects known as immune-related adverse events, or IRAEs.
Which are essentially autoimmune conditions.
That's what they are: colitis, thyroiditis, hepatitis, skin rashes. The development of these side effects vividly reflects how profoundly important those inhibitory pathways like PD-1 are in controlling fundamental self-tolerance throughout the body.
Finally, we need to touch on CAR-RT cell therapy, the ultimate individualized immune engineering tool.
CAR-T therapy is highly specialized and complex. It involves harvesting a patient's own T cells, genetically engineering them ex vivo to express a chimeric antigen receptor that specifically targets their cancer, then infusing millions of these supercharged cells back into the patient.
It's shown tremendous success.
In highly refractory hematological malignancies, yes. But the major side effects are reflective of that massive sudden activation.
What are they?
The two main predictable complications are cytokine storm syndrome, resulting from the sudden massive release of inflammatory mediators, which requires intense medical management, and long-term antibody deficiency, which necessitates chronic immunoglobulin replacement.
Much like a CVD patient.
Precisely. It's a powerful but high-risk intervention.
To summarize this complex deep dive, the immune system is a delicately balanced masterpiece, constantly relying on the crosstalk between the immediacy of its innate forces and the specialized memory of its adaptive forces.
And we've seen how specific defects, whether genetic like CGD or acquired like CV, lead to highly specific clinical patterns. Critically, our understanding of these pathways is now allowing for these sophisticated targeted therapeutics.
From the C1 inhibitor replacement for HAE to the small molecule JA inhibitors.
And the transformative power of checkpoint blockade.
So what does this all mean for the future? The fact that we now intentionally turn off the self-tolerance mechanisms, the CTLA-4s and PD-1s to treat cancer, highlights the exquisite difficulty of immune engineering. The core challenge remains: achieving reliable, aggressive destruction of disease without triggering the widespread life-threatening autoimmune damage that those tolerance mechanisms were initially there to prevent. That ongoing effort to find the perfect therapeutic window is certainly the next great frontier in medicine. For listeners who want to explore more about the primary immune deficiencies we covered, particularly finding detailed information on CGD or SCID, specialized resources like the Jeffrey Modell Foundation, info4pi.org, are fantastic places to start.
Thank you for taking this crucial deep dive with us today.
We'll catch you next time for the next deep dive.